So good morning. Today I will discuss the last part of this unit number 1 and course 15. Now if you have seen your course content, you will find only one part. It is written enzyme catalysis. So because we are dealing with the physical chemistry, and you know that enzyme is a very broad subject. It includes biochemistry as well as the life science and of course physical chemistry as well as the organic chemistry part. So here I will concentrate basically on the enzyme kinetics and catalysis. Now fortunately it is very interesting subjects, and there are large number of good books are available in our library. So I will request to my student that they must refer some of the good books in order to learn complete enzyme catalysis or enzyme kinetics and enzymes. Basically, I prefer this book because you know that there are a large number of books, and even if you see some physical chemistry books like Atkins or Levine, you will get the information of enzyme kinetics. But for today's presentation, today's discussion, I have used this book, that is Enzyme, Biochemistry, Clinical Chemistry, written by the Palmer, very famous book. And this book is also very important, that is Enzyme Catalysis, Kinetics and Mechanism, because I will give trace on the enzyme catalysis or kinetics, and there are some complicated systems. So if you want to learn more about the enzyme kinetics and the catalysis of our biological system, you can refer this enzyme kinetics and the regulation. In addition to that, when I was a student, that time also I referred two books of biochemistry. One is Lellinger, and you must refer the best book of enzyme, I should say, and that is this book is suitable for biology student, this book is suitable for biochemistry as well as the chemistry, that is the Malcolm Dixon, and all the derivations they are very clearly mentioned. Now regarding the course content, because in your prospectus syllabus only one terminology is there, but you can take chair na aapke. But if you see the old question paper or the pattern of our university, so sometimes these aspects are very important. Basically what you have learned, I think, in your school level or your college levels, the most simplest example of enzyme kinetics is Michaelis-Menten equation. Now, on the basis of this Michaelis-Menten equation, we will derive, not today's class, today I will discuss only the one simple system, and my next class I will discuss, because this is very important for biological reaction or biochemical transformation or cell metabolism, everything. This inhibition is very important, and basically there are two types of inhibition. That means you all know that enzyme first react with the substrate, and there is a formation of the enzyme substrate complex. But sometimes what happened in our biological system also, the role of inhibition is very important, and sometimes what happens, there must be some reversible inhibition. That means the activity of the enzymes can be regenerated. But if it is irreversible inhibition, then of course that regeneration of enzymes is not possible. So I will discuss after this Michael-Menten kinetics. I will also try to derive the equation of competitive inhibition. Name itself is a definition. You all know that if I talk about the enzyme, which is a biological catalyst, there are a large number of active centers, active sites. So if it is a competitive inhibition, that means there is a competition between the substrate and inhibitors for that active site. But there are some different types, that is called the non-competitive inhibition, or sometimes it is called the uncompetitive inhibition, and mixed inhibition is also there. So there is no competition for that active center, but sometimes that enzyme substrate complex is affected by that of the inhibition. And then whenever you study about the, or you do some good research, fortunately in our department under my supervision, many students did work on the enzyme kinetics. Basically we use alpha chymotrypsin, we used acetylcholinesterase enzyme, and whenever we study the enzyme kinetics, two things is very important. You all know, that is called the turnover number, the conversion of from enzyme substrate to the products, and it depends upon the active sites, per unit time, and other things. And another is the catalytic constant, and there is a term you all know, that is called the Michaelis-Menten constant, which reflects the activity of the enzyme. So for short answer type question, or for such type of IQ question, you must know some fundamental ideas of these turnover number or the Michaelis-Menten constant. So this is overall our agenda of this course content, that under this enzyme catalysis or enzyme kinetics, you have to learn following aspects, because you all know that according to the new education policy, now it is very important that before studying any subject, you have to know that program outcome, program specific outcomes, and course outcome. That means you must know that why we study enzyme kinetics and up to what level we have to learn the enzyme kinetics. Now it is a mandate of the new education policy. Okay. So let us discuss some very fundamental things of the enzyme kinetics, because sometimes if you define that what is actually enzymes, now it is very difficult for me to tell exact definition of the enzymes, because some enzymes are natural occurring enzymes, some enzymes can be extracted from some plant materials and other things. Some, if you read some books, sometimes it is a nitrogenous substance or like that, but most of the books it is written like that enzymes are biological catalysts. I am just referring this book. The first sentence of this book is enzymes are biological catalysts. Number one. Number two, not most, all the enzymes are protein. So naturally when it is a protein, so there is a very high molecular weights, and basically the globular proteins, more than 60 amino acids and like that. And most important point of the enzyme is the active center, which is very important. And the efficiency of an enzyme can be expressed in terms of K, that is catalytic constant, divided by Michael-Menten constants, and turnover number, as I have already told you. And there is a possibility of, I have also, after this class I will show you some of the question paper of our Pandit Ravi Shankar Shukla University. You will see that there is a possibility of the question related to the turnover number related to Vmax and other things, catalytic constants and other things. And you must know something about the although it is not necessary, because we are dealing with the physical chemistry of the enzyme kinetics. I do not know about your course containing organic chemistry, like that, the classification of the enzyme or the specificity of the enzyme, but we should prepare for all types of the question, because you will appear in the NET, GATE or other examination, or you will do some research in future. So you must know some of the classification of the enzyme as well as the specificity of the enzyme, because you know that enzymes are very specific. Enzymes are very specific. Short answer type of the question. And absolute specificity, all know that if we are trying to catalyze the acetylcholine ester or acetylcholine, which is a neurotransmitter, so only acetylcholine esterase will be responsible for the hydrolysis of that neurotransmitter. So it is a very specific. So sometimes it is called absolute specificity. Sometimes what you know that there is amino acids and large number of functional groups are there. So it will act only on molecules that have a particular specific function. So sometimes we call it a group specificity. Then linkage specificity will act on a particular type of chemical bond. And ultimately, you know, in organic chemistry, conformation analysis is very important, and that is stereochemical specificity will act on a particular steric or optical isomers. And you know that the present status of I must mention the great work of Professor G. Mukesh of IISc Bangalore. Now they are working on the nanozymes, because everybody now we are talking about the material science and nanochemistry. I do not know whether we have included in nanozymes, because some Dr. Manmohanlal Satnami or Indrapal Karwal, we are thinking to do some work on the nanozymes. It's very recent work of science, nanozymes. Okay. Now there is some confusion about the history of the enzymes or history of the catalysis. You all know that Berzelius discovered the catalysis and Buchner and so many things. But whenever we concentrate only on the enzyme kinetics, so you must know that basically we always consider that Michaelis and Milton, two different scientists. One is from German and another is from other countries. Actually, we consider that they actually started that enzyme kinetics. In some books it is written 1913 or 1912. They have published many papers during this 1911 to 1913, but this is the first paper of, so therefore we always, to bore their name, we always consider it a Michaelis-Menten kinetics and Michaelis-Menten constant. And but we must consider the great scientist Victor Henry, who studied large number of sucrose hydrolysis type of the reactions. And he, I think, first enzymatic reaction is one type of fermentation and like that. But they have used some urea, hydrolysis of the urea and other things. So as far as the origin of enzyme kinetics, we must mention the Michaelis and Menten, as well as, of course, the Victor Henry. And this is the great picture of this great scientist L. Michaelis and Madame Maud Menten. And actually in 1913, they published their seminal work, fundamental work on enzyme, almost all of which are proteins. So that is the, I have taken this photograph from this, the science history of this enzyme catalysis. Even if you want to learn about the many things about the catalysis, last year Nobel Prize onto the organocatalysis, and if you read the Nobel lecture of this organocatalysis, you will get all the information about the history of the enzyme. But our objective is that to derive the simple enzyme kinetics. And student must know some of the very simple example of enzyme kinetics. And I am not talking about the classification and other things. You all know that the hydrolysis of the urea or some sucrose into glucose and fructose. So name you know that invertase, or like if we are doing the urease or like that. These are all, but we must know some of the examples of enzyme catalysis, early example of the enzyme catalysis. And I always give this example to my student because we have done a lot of work on this. Even still also Dr. Manmohan Lal Satnami of our department is also working on these things. So because it is one of the most important biological example of the enzyme kinetics, you know that it is acetylcholine, which is a neurotransmitter, and when it will hydrolyze, it will form the acetic acid and choline, and that enzyme is responsible for this hydrolysis. That is called the acetylcholine, which is the most important biochemical example of the enzyme kinetics. And we have done a lot of work for this type of the reactions. And if you read some of the good books and the research paper, you will get complete information about the turnover number, which is the most important characteristic of an enzyme, and then Michaelis-Menten constant and other things. So let me start with this first simple system, and then of course, if time permits, then we will discuss about the competitive inhibitions as well as the uncompetitive inhibition, as I have already told you, and non-competitive inhibition, mixed inhibitions, and how these inhibitors affect this Michaelis-Menten constant. And if you are working on some hydrolysis reaction, suppose you have taken some succinic acid like that, and you are using succinate anhydrous enzyme, normal catalysis, and you add some malonic acid or some things. So you know the structure of this, both the substrate and the inhibitor is almost same. So in such case, that part inhibitor suppress the activity of enzyme. So that is called the competitive inhibitions, and these examples are also available in all the books. And this is the most simple example of this competitive inhibitors. And uncompetitive or non-competitive inhibitors, sometimes the action of pesticides, or sometimes the heavy metal pollution, they will attack some of the group, sulfohydryl group and other things. So that is non-competitive inhibition. So let me start with this. I will not discuss today this thing. I will first discuss the normal case, and then I will problem some numerical problem, and then. So now one thing I should tell you that, okay, today I will discuss only the Michaelis-Menten kinetics or the simple system. But you know that in M.Sc. first semester, you have already studied the steady state conditions, and or sometimes it is called the steady state hypothesis. But you know that if you, as I always told my student, that if you understand one system, after my class you must see some of the standard textbook or some books related to the enzyme. You will find there are a large number of equations, but always we prefer this first equation, but that Michaelis-Menten equation can be modified. There are so many examples are there. Like another very important example is called Lineweaver and bark plot. Lineweaver and bark plot. That is sometimes we call the extension of the Michaelis-Menten kinetics, and ultimately we get the velocity of that type of enzymatic reactions. And you will be happy to know that sometimes enzymes are commercially available. We do purchase alpha chymotrypsin from sigma LV. We do purchase acetylcholinesterase, electric eel, but if you want to do some work on the human acetylcholinesterase, then that is a very difficult task. From blood, we have to extract that human acetylcholinesterase. It depends upon your researcher. Okay, so let me start the first equation, which is very simple, and I will first solve it and then I will do all these things. And it is very simple to understand. So today I will deal with the simple system. Simple system. Sometimes it is called, as we have already told, Michael-Menten kinetics, or the determination of K n. So suppose E is the substrate. I am not showing you the structure of an enzyme, how it reacts with things. Suppose there is a part of, I am not discussing that very old school level key and lock system and other things. I am just simply using physical chemistry concepts. So E plus S is the substrate. And you all know that the concentration of enzyme is very, very low and the concentration of substrate is high. So there is a equilibrium state and formation of E, enzyme substrate complex. And as usual, your normal kinetics, will write K1, rate constant for forward reaction, same thing, this is enzyme. And then enzyme substrate. A slow step product. So, and we did a lot of work in our lab. So basically we can write like that: rate is equal, as because this is the slow step, so yes, it is very simple. But we cannot ignore this equation, because whether it is a competitive or non-competitive, anything, you have to apply the same method. If it is a competitive inhibitor, so there is a possibility of E I, so E I, so another possibility. So we cannot ignore these things. It is uncompetitive, so, and what should be the effect of like that? ESI. So therefore, first we have to learn this and then gradually these things. Now applying steady state approximation. So we have to determine this. So yes, very simple. So K1 minus again minus concentration. Only one step. As because it is steady state approximation, it becomes 0. So we can write like that. K1 ES. Or directly you can write like that. Or directly you can write like that. So very simple. Now the last step. So we can substitute the value of ES from here. But before that, we will do one thing, that just like the normal first order kinetics, what we do, suppose we think that the initial concentration of starting point, and some enzyme reactions are very fast, some enzyme reactions are slow, but we assume that there is a possibility of now it is not necessarily like you can write like that, you can also write like that in some books. If you see this, so I don't mind it, you can write like that also, or even you can write digital. What I mean to say that, so both are correct. This is also correct, this is also correct. It is up to you. So I can write like that. E0, whatever I write, so it is not minus E. I am taking the book of the Ladler like that. So you follow my point. You can write like also, you can write like that. You substitute the value here. So I will get K1 in place of this is E0 minus ES S so if I just write that K1 0S because K1 ESS. Again, I can in some books, because substrate in comparison to enzyme, the substrate concentration is very high. So in some books it is written like that also, but it makes no difference. So it is up to you. That means either you can write S or S0. So that is not a very important point to be discussed. So in the last step, that we have to substitute the value of ES from this equation to our main equation, K to ES. So So last point is that rate, now K2 as usual, K1 E0 S actually Michael, if you read the biography of K1, if you read the biography of the Michaelis-Menten research paper or the history, they have done a lot of work, and sometimes it was thought it was empirical type of the things, but ultimately they got the final results for that. They thought that suppose both numerator and denominator is divided by K1. So change the color, that K2 E0. Again, you can write here also or outside. S K1 plus K2. Deliberately I am writing in the red color. Again it will yes. Now according to, I'll explain, and you must write some of the values of this. According to Michaelis-Menten, this red color, that means K1, K2 and K1 is regarded as Michaelis-Menten constant. In some books it is written like that, M, or you can write, that is not a problem, small m also. So it is very, very important reflection of any types of Michaelis-Menten constant. Now sometimes there is a constant. You will see some of the values of the Michaelis-Menten constant. So in this place, now the role of inhibitor is there. That means suppose I add some inhibitors, then what would be the effect of that inhibitors on this Km value? A short type of question or objective type of the question. Should the Km value increase or decrease? Now on the basis of this curve you can plot these things. So I can write now the K. Now what happened? Suppose the concentration is very low sometimes, substrate concentration, and as compared to Km value, this K is maybe neglected. So that time rate is dependence on this S. So sometimes there is a probable question, and if the concentration of S is very high, concentration of the substrate, as I told you in some books it is written like that S0. So that is not a important thing, but if it is very high substrate concentration, then of course that Km value can be neglected. But of course everything depends upon the experimental condition. if you do some very simple experiment, spectrophotometric experiment like acid catalyze or alpha chymotrypsin hydrolysis of methyl acetate or some para-nitrophenyl acetate. Generally, we do some paraxon on parathion. Then, of course, we are able to correctly tell, yes, sir, the value of K1, K inverse 1 or K2 like that. So, then, of course, this so it is sometimes it is called the zero order, independent of the substrate concentration. So sometimes it is called the zero order kinetics of light. So they have if you see, as I already told you, if you read that research papers of the Michaelis and Menton, so they have done many experiments, and on the basis of that experiment, they concluded that yes, there may be possibility of zero order reactions or the first order reactions. Now the question arises that the last part of this reaction, this type of reaction is actually called the Michael-Menton formula or other things. Now on the basis of this, there is a modification, large number of Eddy system and Haldane system and Lineweaver and the Bark system, because sometimes what happened, that means now if I want to ask you that how do you calculate the Michaelis-Menton kinetics. So what they did, Michael and Menton, they plot a graph. I am just showing that graph. They plot a graph between the rate and that substrate concentration. So the last part. Why it is important? Because if you understand the simple system, then you can solve any complicated system, because the methodology is exactly same. Only thing is that you have to consider the value of Ki. You must include the value of Ki in our equation. Then K, EI, ESI like that. Yes. So that cannot be ignored, and then we'll plot. So if we plot a graph between the, I am coming to that point, rate and S0 or S, as I told you, in some books it is like S. So such type of, I'll come to this point. And we have seen a lot of work in our lab. So there is a constant value. So we always call it is a Vmax. And we can write like that. This sometimes this K2 is 0. Actually, this is actually B0. Vmax we can call it. Vmax we can call it. But again, although yes, we can determine the Km and other things, but precise determination of Michaelis-Menton constant is not possible. Of course, now we have a software, others say parabola, or we can determine these things. But precise determination of Km is required. I will ask you one question after this class. If you are able to solve it, then I think that yes, you have got my point. So this is the simple system. Now the problem was that actual determination of the Km, which is Km is the characteristic of any enzymatic reactions. I will show you some of the results of the Km and catalytic constant values. Say for example, as I told you that we are dealing with that acetylcholinesterase and the turnover number is 10 to the power 4, 10 to the power 4 to 10 to the power 5. Can you imagine the turnover is very active catalyst. Now the problem is that to rate or we large number of modifications have been observed in the literature, but one of the most important and most useful modification was proposed by two scientists. One is Lineweaver, very important for examination also, Lineweaver and Bark, Pink Bark. Very important modification. Not modification, I should say same thing, just a double reciprocal plot, that is 1 by rate like that. I should not say, but they modified it. So we always remember their name, the Lineweaver and Bark. Lineweaver and the Bark. But whenever we do any type of enzyme kinetics, so we always determine the Michaelis-Menten constant with the help of this Lineweaver and Bark plot. That is nothing, it is slightly modified. The mathematics students can, that is called double reciprocal plot. That means you can reverse it, Lineweaver and Bark plot. That means if I write 1 by rate, just I reverse these things. So it is Km divided by K2 E0S. I'll write E0S. No, K2 is 0. K2S. I think let us solve it first. So no S is required. Now just I write in order to, I'll write here, that is better, because I will ask some numerical problem. So this is Michaelis, and second, again I will write here, no problem. 1 by rate, same thing I will write again, because Km, now my question is that Km, K2 E0 S. Okay, you can solve it. Plus, easy, no S. Now, you just solve it, you will get these things. Nothing to be. Now my question is that, what is the difference between these two? That is called the double reciprocal. Now, my question is that, if we plot a graph, means Lineweaver and the Bark, they actually plotted this graph, 1 by rate or 1 by V0, you can write, and 1 by SG. S or SG, whatever it is, as I told you. Fortunately, it's a double, this is simple mathematics, double reciprocal plot. Straight line is outside. I'll show you that plot. So you know that y is equal to my last question. X, so slope is equal to Km slope and intercept okay. So this is normal Michael-Menten curve, V maximum you can write this thing. Here this is intercept, whatever it is. This is slope. Now my question is that, just you listen very carefully, my last question, then I'll show you of course that what is the relationship between Km and K0 analysis. How do you calculate the Km from a line over and bar plot? How do, first you apply this Michaelis-Menten kinetic, okay? You just reciprocal, double reciprocal plot, 1 by rate, same with the same data, just you reverse it, 1 by rate and 1 by initial concentration of that substrate. Different substrate you have taken, you are getting 5 points, and with the help of computer you are getting some slope value, and slope value is equal to Km divided by K2 is 0, and intercept value 1 by K2 is 0. So my simple question is that, how do we calculate the Michaelis-Menten constant from this plot? Anybody? Just think for 1 minute. Anybody? Ayushi Parmar or any Devangan, Vikas, just you slope divided by intercept, slope Km0 divided by 1 by K2, K2 is cancelled, so you will get the Km value. So just division. Anyway, now let me just see the slope divided by intercept. I'll give one question, then I'll discuss these things. Basically what happened, although I am not saying that with the help of Michaelis-Menten equation one cannot determine the K, but precise determination of K can only be determined with the help of line waiver and the bar plot, and there are others plot also. I'll explain step by step. So this is normal case. Now see, same thing, 1 by, this is line waiver and the bar plot, so slope is equal to Km and V max, same thing, K2 is 0 like that, and intercept is 1 by K2 is 0. So just if I divide slope divided by intercept, we will get this value. Now actually, that why this Km is important, because this Michaelis constant is an indication of the affinity of the enzyme for its substrate. We must know the Km value, and it is also a characteristic of a particular, just like if we do some normal equation, we are concerned about the energy of activation like that and other things. So similarly for enzymatic kinetics, one must know the Km value, because it is we must know the enzyme substrate complex. And if the value is smaller, then the more strongly the enzyme binds the substrate. And you see the range of these Km values. I will not show you the range of some of the values of some of the Michaelis-Menten constant, but Km values range mostly lie between 0.1 to 0.000000 m. So this is the value of Km. Third point is that at very low substrate concentration, when S is much less than Km, so we can calculate the V max. You already know that the rate is directly proportional to the substrate concentration. I have already discussed these things. And if the high substrate concentration, so it is independent of that substrate concentration. From graph also you can determine. Now the last point is that whenever we study any enzymatic reactions, so Km is also important, but one very important part that is called which is another important part. If you do some research in the enzyme kinetics, so you always prefer that your K catalyst should be very, very high. The value should be very high. So therefore, and this, again, it is a probability of the numerical question in your examination or like that. How do you calculate this K catalytic? So K catalytic can also be calculated with the help of course, V maximum, again that is K2 is 0 and divided by total enzyme concentration. So you can get that this value of the K catalytic. And turnover number is also very important, and the value of this turnover number is the 1 into 10 to the power 4 per second. Maybe high, less than that. And as I told you in my starting point, that all such type of Km value or the K catalytic values and turnover number is based on, or the application of this part is to understanding the cellular metabolism as well as some physiological diversity. So thank you very much.